Good morning. Okay, I know what you're thinking. Man, Andrew sure looks different this morning. What happened to him? He can't be with us today, so he sent me instead. For those of you who don't know me, my name is Didi Ortiz, and I am the Senior Program Manager for Science Education here at the lab. I have been here for about 15 years and managing this particular program for about the same amount of time. So I think I'm a lifer, like most of us are here. I would love to begin with the usual housekeeping. Welcome back, as always. And because Andrew can't be here, he sends his love through bagels and donuts and coffee, as he normally does. And yes, please silence your phones so that we don't interrupt Professor Lockwood's chat. And please hold all your questions till the end. Once the talk is over, you can queue up by the microphone stands and we'll just go zipper style to answer questions. If you're on Zoom, please make sure you are on mute and you can use the raise hand function to ask your question, or you can type it in the chat and I'll read it afterwards. We are recording the talks, so in a couple of weeks, once they're edited, they'll be posted online or on our YouTube channel, I think it is now. And as I allow you to peruse Dr. Lockwood's bio here, let me introduce you. Please meet Julie Lockwood from Rutgers University. Thank you for being here with us today. And as usual, Andrew style, we're going to ask her a few questions. The first one is, when do you think that you were interested in science? When did that hit you, that curiosity?
So just confirming that everyone can, yes, good, okay. So, so I have to confess that my youth was dominated by sports. So I grew up outside of Atlanta, so I still have a little bit of a Southern accent that will come out. But the social currency for any of you guys who ever spent time in the South is sports, especially football. And I grew up with a dad who played a lot of sports and two brothers who played a lot of sports. And so that dominated my life. So I didn't realize I cared anything about science until I was a senior in high school.
You're singing to my soul.
And that was almost by accident. So sort of in a quest to make sure I wasn't in a lot of classes in my senior year in high school. I took some classes at the community college next door. And the only class that fit into that schedule was an advanced biology class. It just fit, so I took it. And I really, really liked it. I had a great teacher, thinking of that, and she really kind of turned me onto it. And so by the time I went to college, and I chose my college based on a scholarship, not on academics, a sports scholarship, was I'll just major in biology. I really liked it. And so I just kept following that gut feeling, and here I am.
Excellent. So, perfect segue. Was this the only person who inspired you to continue in the field, or was there somebody else along the line that said, hey, I might like this?
So, there are several people along the line that kind of made a big difference. I mean, that high school teacher was a big one, even though I never really got a chance to ever tell her that.
Might be watching.
She might be, although, I'm old enough. That's maybe not.
29.
So, and then when I was in college, another happenstance circumstance landed me in really a really key mentor's lab for my senior year in college. And that got me started. I ended up doing a master's degree with him and carrying on. So his name was Mike Moulton. He's at University of Florida now. I think he's retiring. But that was my key moment. He kind of felt sorry for me and went forward from there. I had done some stupid stuff, and so that's and he helped me out of that
Shout out
jam.
to Professor Mike.
Yes.
Okay, so besides biology and research, what else do you do for fun?
Right. So, again, sports, right? So that hasn't changed. I head out in a couple weeks to meet my brothers, and we watch the SEC women's basketball tournament in South Carolina together. And we just some time to break from our families and just sit around and eat Reese's cups and drink Coke and watch basketball. Other than that, I like to canoe and go for hikes. We spend a lot of time in the Adirondacks, up in the high peaks for those people who may go up there. So that's one of my favorite places in the world. And so we spend a lot of time up there exploring and try not to lose a lung trying to climb those mountains.
Kind of shames me into my 7 steps per day. Okay, bonus question.
Yes.
I'm sure we are celebrating our 75th anniversary here at the lab.
Yeah.
I can't imagine how much the biology field has changed in 75 years. Do you have an idea?
Well, so my segment of biology is ecology and evolutionary biology. So biology split actually a lot in the 70s and 80s, according to more of a biomedical point of view, and then this kind of, or being a big part of the Earth system's point of view. So ecology, its roots go all the way back to Darwin and Alfred Russel Wallace and these sort of really talented natural historians. And so that dominated the whole field up until the 50s and 60s. And at that point, ecology kind of, what they call it physics envy. And they thought, hey, you know, we can move into a more of experimental and theory-driven and hypothesis-driven science. And so that was the big shift, is in that period of time, it moved from observations into experimental design and hypothesis testing. Since then, it's sort of balanced out between the two, especially with the advent of big data, large data, environmental data sets, and big problems like climate change that are not necessarily amendable by experiments.
I wonder where we'll be 75 years from now. All right. Well, thank you very much for the lovely introduction, and the floor is yours. Please welcome.
Okay, thank you. So forgive me while I do the Zoom shuffle. Okay. So what I'm going to talk about mostly today is biodiversity and paint rollers and DNA. And so I'll weave those all together and start and sort of introduce you to a DNA-based revolution in how we track biodiversity. So that's my ultimate goal today. Oh, I forget. You have to go there. So for those of us in the room, remember these first pictures of Earth from the moon and how startling they were and how much it put our world into perspective, right? So this is a quote from one of the Apollo astronauts, and this is Earthrise from the moon. And, you know, that sort of sentiment that he is passing on is once you move from the face of the Earth and out, you realize how fragile this place is. And in particular, when you look back on Earth, what you dominantly see is just blue, right? The blue planet. This is sort of the generation of the whole idea of we live on a blue planet. If you look just at the hard parts of the Earth from space, then you'll see some green and some brown in there too. But what I want to sort of point out here is that, you know, starting from the biggest perspective, that when you look back on the Earth, you realize how fragile this place is. And in particular, when you look at the Earth, what you're seeing really is the biosphere and the thing. And what you're thinking about is the stuff that's living here, because you live here, right? So if you move a little bit closer into the Earth and you say, okay, that's our Earth, what is it made of? So this is just a cross-section going from the core of the Earth to the surface of the Earth. What all my Earth system colleagues will point out is that most of the Earth is hard, right? It's the geosphere. It goes from the inner core, which is not so hard, but if you left it and let it cool, it would become hard, right? All the way out to the continents that drift on top of all that. And then, of course, the part of it that it peaks up and becomes our hard surfaces, landscapes, mountains, etc. So when you look a piece of that Earth, you realize one of the things that you realize is that the living part of the Earth, the biosphere, is a tiny, tiny little sliver of the entire Earth's surface, right? So the part that represents the living part is anything on top that's in the water or exists in the atmosphere right along the surface of the Earth. It represents a 0.0007% of the entire Earth volume. So if you were to take the Earth and scale it down to the size of a basketball, which of course I just told you I love, is that the varnish on the outside of the basketball would be the width of the biosphere. So that's it, right? That's all of life. All of life exists in this teeny tiny little shell around the edge of the Earth's surface. All the things that live in that living part of all the species that live in that part of that little thin veneer of the Earth's system holds between 1.5 to 2.5 million species that have been formally described, and then an unknown but very large number that have not yet been described. It's a real trick to take what we know and then extrapolate to what we don't know. So we have like a big range in terms of people estimating how many species are really on Earth. going from around 8.7 million up to about 1 trillion. So that's all life, all species on Earth. All of them live in that little thin thing that we call the biosphere, right? All of which is floating around in the giant expanses of the universe. So this is a representation of the evolution of life on Earth. It's not necessary, of course, that you be able to read all those parts. You're supposed to be a little bit impressed by the size of it. But the way you read it is the very middle of it is the generation, it's the first single-celled organisms from which all life has descended about 3.5 billion years ago. And then you can think about that in a different space as being the root of the evolutionary tree. And then from there, you have all species kind of evolving out to make this beautiful tree, right? So it's representing that tree starting from the core, the first signs of life on Earth, all the way out to today. Each one of these lines represents about 500 different species. So this is not just any one species along that branch. This is about 500 of them. The inner ring represents everything that we have already described, and the outer ring represents some extrapolation of that to understand what it looks like. So there's a couple big take-home messages here. Like the big tan part is all the arthropods, the insects, the crustaceans, and spiders, right? So most of the described life on Earth is insects, right? Another big chunk is the bacteria and viruses, which we know pretty well as well, both good and bad. And then you take a look at the little green part, of course, is plants. And then the part there where I say you are here are all the vertebrates, sea stars, urchins, and things that you classify under the Deuteronomy. And you are there. That's you. You're one of those lines. You're one in 500 of those species on one of those lines, right? And in fact, it's not a very deep branch. That our branch is really, mammals have been around for a long time, but our branch of mammals is relatively new, right? So this is a piece of just understanding the biosphere and the world in which you share with other species on Earth. And most of the species that you're sharing on Earth turn out to be insects and bacteria and viruses. If we just blow out the chordates, which includes all the things that you can think of that are the vertebrate animals, the fishes, birds, amphibians, reptiles, mammals, and a few things that you throw in there like the agnatha, which are very early fish, jawless fish. So that's, I'm blanking on what they are right at this moment. There's some good examples, like in the Great Lake. Oh, lampreys, and a really cool, and hagfish. See, I knew it would come back to me when I don't know if you've ever seen videos of hagfish, but they're very cool. They live at the bottom of the ocean. They're like the detritivores at the bottom of the ocean. They're very fascinating. But that's a very ancient lineage of fish. And then a couple of things that have some characteristics that are like pre-vertebrates, like the tunicates and things like that. Even when you blow that up, that part of the whole tree up and look, right? We kind of sit in the mammals, and we're not even that common in terms of all the mammals. Most of the vertebrates. Most of the vertebrates on Earth that we share this world with are fish. So there's about twice as many fish as there are every other vertebrate, right? So I taught vertebrate zoology for about 10 years, and I had to say, we could just spend 90% of the class talking about fish. And the big key thing about the fish is there's one little line in the fish that gave rise to all the terrestrial vertebrates. So if you're a fan of Nova and nature and all those things, and you've heard about Tiktaalik, that was one of the early fossils that we show that came from water onto land and probably was one of the early examples of terrestrial vertebrates. So we're all fish at heart, right? So one of the things that I spend a lot of time thinking about is that all this fantastic diversity that we share the Earth with is under some sort of stress because of us. So this is a graphic that was produced by the International Panel on the Biodiversity and Ecosystem Services. So that's a UN-level worldwide consortium that's equivalent, for those of you who are familiar with, the International Panel on Climate Change. So these are the two big panels that are organized every year, every other year, to kind of give an assessment of the entire Earth system. What climate change is clearly climate and the pieces that are changing, and this is related to biodiversity and its services it provides to us. So I'm going to start on the sort of left side of this. So they have divided this up, and anytime you see the percentage there, that's kind of the percentage loss. So it's a very kind of negative start out of what have we lost relative to certain ways that we can think about biodiversity or nature in general. So the couple of big ones are the sort of extent of natural ecosystems has declined across the world by about 50%. So what came in its place? Urban areas, agricultural mostly. Species extinctions, about 25% of all species that are described are at risk of or already extinct, like in modern times, not the ancient fossil extinctions. A big one is biomass loss, and just loss of like numbers of individuals per species. About 82% of wild animals, and this is just looking at, this is they can only measure from like vertebrate animals, for example, and maybe for insects. That total biomass, total amount of sort of insect bodies and vertebrate bodies that exist on the face of the earth has declined by about 82%. So if you've heard of the insect apocalypse, have anybody heard of that and read about that in the Times and stuff? So that represents that idea, that through a lot of the drivers that we see here, the number of insects in our world have gone down. Another thing that can kind of bring that home is that, especially me growing up in Atlanta, we would drive to Florida every year, right? You have to go to the beach. And as you're driving through Florida, you get about halfway through and you have to stop and you have to get that little squeegee thing out because there's so much insect crud on the windshield that you couldn't see. Okay, that doesn't really happen much anymore. That's an example, and kind of a real life example of what it means by that loss of biomass. We've just lost a lot of different individuals. Populations are not as big as they used to be. And then another key one is that nature, which I'll talk about in a few minutes, has profound meaning to people, either just on an individual level or a cultural or social level, and especially in regards to how indigenous communities interact with nature. It can often have a connection to the origin story. It has a connection to a tradition that's been passed down into families. And so that sense of loss from the decline of some of these species is when you ask these communities to report whether or not they have felt a nature loss, then fully 70-something% of those respondents would say yes. So what's driving all this? The direct drivers are the next thing over here. Those are landscape and sea use change. So that's, around here, you feel it every day, or you see it, I guess. And the level of urbanization, it's like, oh, look, another Walmart, another Costco. So what did it go over the top of, right? If it was an old Walmart, I suppose it doesn't really matter in terms of this sort of question. But if it took down a forest, then that's a land use change, right? And seascape, it's a little more subtle because we don't swim in the sea and we don't see a lot of it. But certainly in terms of some of the fishing we do and the tactics we use in terms of fishing or deep sea mining, then that seascape also changes. There's direct exploitation. That's not food production necessarily, but that's fisheries. It's trophy hunting. It's the capture and sell of species that we like to keep as pets that are not cats and dogs. So think about the geckos and lizards and birds that you can sometimes see. Those are all direct exploitation issues. Climate change, this is one clearly that's increasing, that as the climate warms in general but becomes weirder. I should say you should call it climate weirding and not climate warming. It just gets bizarre and changes the phenology and a lot of other things that species have evolved with. So if it changes too much, those species will they'll often decline in population. Pollution, certainly invasive alien species. So these are species that have been moved from one location to another with the help of people. And not all of them, but some of them will go on to cause pretty severe ecological damage. For example, there's a lot of island birds that have become extinct over recent 100 years or so. because of the introduction of predators in places where there were no predators before. So all those little birds and stuff just are like completely naive to any kind of predator, and so they're like, oh, hey, you know. So in those cases, that's where you see a lot of these issues with the invasive species. Not so much, I would say, here, but in some sort of really vulnerable ecosystems where there's a lot of very special species that you can find nowhere else, you often see these issues with invasive species. We include in this category of invasive species emerging pathogens. So for those of you who are familiar with amphibians, and this one does affect us here, there's one, and reptiles, there's been two pretty nasty pathogens that have been introduced, probably from the pet trade, and then have spilled over from our aquaria and everything into our wild populations. And again, they're very naive to these pathogens and they die off pretty fast. So that all gets clumped in there. And then there's the others category, right? So these all drive back to socio-ecological or socioeconomic drivers, which we won't go through, but I kind of want to set the stage of these are the things that we're doing to the other species that share that little thin bubble, that we exist in the world. One more doom and gloom slide, and this is another product out of the International Panel for Biodiversity and Ecosystem Services, but this is taking data that's produced from the International Union for the Conservation of Nature, the IUCN, and their goal is to go through and pull a lot of experts together, evaluate whole groups of animals that's reasonable for this group to deal with. It can't be too big. And then go through and take all the evidence that we have and all the data we have and put it through a very kind of focused, systematic process to say, is this population likely to go extinct? Is it close to going extinct? Is it extinct in the wild, which means there's nothing left but things that are in botanic gardens or zoos? Or are they doing okay, right? And so they'll do, anything in green here is the doing okay. Through a really systematic look at the evidence, they're doing okay. Anything in red or that kind of red color is stuff that they would classify as not doing great, right? Anything in gray means we just don't have enough data to know. So these are the groups that they have managed to go through over the span of 20 years, I think. Sometimes they'll update these to sort of keep what's going on. And the biggest group you see are cycads. I don't know, how many people know what a cycad is? So these are ancient, ancient trees. They're like these leftover trees, from long, long ago. They look a bit like palms, but they're not even remotely related to palms. You're not going to find them here unless you happen to be in a botanic garden that has one, and they probably keep it inside around here because it's too cold. But they're native to, you see there's a lot of them in the Caribbean and northern South America and Southeast Asia. And the place I think about them a lot is in South Africa. So they exist in these, they can be kind of arid, but they exist in these sort of tropical, subtropical areas. Very cool trees if you want to look them up later. But they're not doing very well in today's world. There's only about 300 of them left, so you keep track of that number along the side to give you a sense. After that is amphibians. So the amphibians are also not doing well. In part because of those disease and pathogen spillovers, in part because they're like the canary in the coal mine for a lot of the things that happen to ecosystems. They're very vulnerable to some of those. They're also turn out to be very vulnerable to climate change. It's just part of their life history, and the fact that they often, like the ones around us. So if you don't know, then we live on the Appalachian Mountains and the whole chain in a hotspot, evolutionary hotspot for salamanders for the whole world, right? And we have a whole bunch of salamanders here that breathe through their skin only. And so that's a good example of like when a little critter just breathes through its skin, anything that affects its skin is going to kill it, right? So if you were a salamander junkie and you in the whole world and you wanted to see cool salamanders, you would go to the mountains of Tennessee. If we're interested in marsupials, right, we go to Australia, maybe to South America. So that's our claim to fame in North America, are those amphibians and freshwater mussels, but I won't talk about those too much. But But that kind of gives you a sense of why the amphibians are kind of in a little bit of a dire straits. There are other groups, of course, but you can see that like the balance of green to red. The last thing I'll say is that you can see there's a lot of gray in some of these areas, which means we just don't know. No one's looking. So there's a space here to sort of increase our ability to even just understand what's happening to different species. Okay, no more doom and gloom. From here, it's all hope, right? So I often asked, that might be a southernism in there, sorry. So I don't realize I'm saying it, and my youngest son was like, what did you just say? You said fireworks? I didn't hear that. So this is another conceptualization of why all these species matter, right? And so that's a question that I'm asked. It's like, okay, things are declining, so we lose a little frog. Who cares? I mean, does it really affect my life, right? And so this is sort of a graphic that explains how everything about it is what we do, everything about what we do is connected to the species we share this earth with. And these are often labeled under the term ecosystem services. So I always start with this one thinking about like a completely impossible possibility, and that's that the whole world is just all the water and rocks, and it's just us in life. There's nothing else alive. So clearly we can't survive, right? Why not? Because other species in the world provide our food. Other species in the world provide the oxygen that we breathe. Other species in the world provide the medicine that we create, or at least we discover. Other species in the world move the water and clean the water. Other species in the world pollinate all our crops. Other species in the world help create the raw materials that we use. Other species in the world create soils that are fertile and can grow our crops or to do the things we want to do. So there's some very fundamental things that we rely on all those other species to do, right? There are other pieces that are maybe not as fundamental, at least on a like I need to live and eat scale, but are still important, including climate regulation, flood control, a lot of the forests around us that go right up to the rivers do a lot in terms of ameliorating the floods that are becoming more common as the climate weirds, as climate changes. Especially all the forests that we have up in the northern part of the state or anywhere in, up in the Delaware watershed, keeping those forested reduces the scale and the flashiness of all the floods that we have. So keeping trees on the landscape in our area is very important, and all the pieces to that. Then there's the aesthetics, the spirituality, the recreation, the things that are a little harder to get your, to put a dollar sign on, but those are the things that also make us happy. So when I go to the Adirondacks and I say it's my favorite place in the world, like what am I responding to? Well, I'm responding to the mountains, and I'm responding to things like that, but I'm also responding to all the nature that's around me, and it feels good, especially leaving New Jersey, to go there and experience that. So nature gives us that as well. So what are we doing? What are the things, how do we describe what it is that we do to sort of keep that natural and those ecosystem services flowing? There's three ways we do it. One is through conservation, one is through preservation, and the other is restoration. So these have roots, historical roots back, and I can talk about those if you want, but conservation is protecting nature through proper use of natural resources. It's the idea of if we need stuff now, like you can't live without energy, right? We can't live without food. So those things that we need, can we derive those from nature in a way that we don't deprive future generations of being able to do the same? So that's the idea of conservation. Preservation is protecting nature from any use or interference, right? So that's captured in national parks for us. National parks, some cases in the state level as well, or even local. And then restoration is taking a natural landscape that's been degraded for whatever reason, and then using all our knowledge and tools about how nature works to recover it back to something else, something a little more natural. So that's restoration. In New Jersey, that's sometimes taking brownfields and making sure they're not toxic anymore and returning them to something at least you want to take a walk in. Maybe you don't want to eat anything out of there, but at least you want to take a walk in that. Or taking wetlands that have been kind of destroyed for one reason or the other and recreating the right hydrology so that you have all the things that are supposed to be in a wetland. And it serves the functions that we need it to do. So this kind of gets us around to Aldo Leopold. I'm sure most of you have heard of Aldo Leopold. If not, you had to probably read it in high school or some level, right? But this is a quote that I want to bring up. This is a quote from Round River. So the last word in ignorance is the man who says of the animal and plant, what good is it? If the land mechanism as a whole is good, which is this idea of ecosystem services really, then every part is good, whether we understand it or not. If the biota in the course of eons has built something we like but don't understand, then who but a fool would discard seemingly useful parts? To keep every cog and wheel is the first precaution of intelligent tinkering. And I want to talk from here on out about that idea of every cog and wheel, right? So every cog and wheel, what he means there is the species that we share the earth with. So they're distributed in ways, they interact in ways that we don't often understand, but we certainly benefit from. So how do we keep track of every cog and wheel, especially when I showed you how many species there are in the world, right? So I want to talk now, this is sort of a question, so I'll have to tell you that about 10 years ago, probably no accident of the fact that my kids were, I had emerged finally from babyhood and like having the fog of having 2 little kids and being like, I'm a little worried about their future. Like, what is this going to look like? I know what's happening. I understand what's going on. I sit in this space where I'm paid to learn and understand these things. So what can I do, right? Society has invested a lot of me to become a well-trained scientist with a good job. So what is my responsibility in terms of finding solutions? So I was just told my lab was like, solutions, solutions, solutions. We're only working on solutions. I'm not describing the problem anymore. We understand the problem. I want to go towards solutions. And from doing that, one of the things I realized is this every cog and wheel problem is substantial, right? How do we keep track of all the species? The way we have done it in the past is from things like this. And I'm just going to review these a little bit. The first one up there is keeping track of fish. So you have to catch the fish, you have to get them on board. If you want, hopefully you didn't kill them in the process, but sometimes that happens. And then you like, how big are they? Are they reproductively mature? Are they males or females? And then you can put a little tag in them. These days with technology, you can put a really cool tag in them, and then you can throw them back in the water and figure out what they're doing. A lot of really cool information, but really intensive and kind of one species at a time. Here's another one where they're flying these surveys for big things like elephants. In that case, so I haven't surveyed for elephants, but I've surveyed for birds in those airplanes, and I'll tell you what you're doing up there is that you're sitting like this, looking over on your side, trying to record what you see and not throw up. So, you know, you can't really record everything. You're just looking for, you have like the search image for these birds, and you're like, okay, it's like over 1,000, and I'm writing it down, and you're like, oh, okay, please. So it's expensive, time-consuming, on you go. Sometimes when you have to, you know, you have to pretend like you're a fish and get under the water and record what you see there. For insects, you take advantage of the fact that this is a moth trap. You take advantage of the fact that moths are attracted to light. You put a light source out there, you set things up, they fall in, and you count how many things that you caught. Going through, and the only last one here I'm going to do, you can't really see it, but right up in their little corner is a little snake, I forgot what. Anyway, there's a little snake right there, and what this getting pulled up is what's called a cover board. And so what you do to survey for these things that you're just not going to see very often, and some of you are probably really happy that you don't see snakes very often, right? But when you're trying to find out that cog in the wheel, it's really hard. So what you do is that you make a little microhabitat that makes it really, that's very makes snakes really happy, snakes and lizards. And that's just by putting a board on the ground and leaving it there. And it creates a little nice moist place for them to hang out and sort of safe from predators. So you put out enough of those, and you go up to each one, and you very carefully lift it up, especially if there's a poisonous one in the area. And you make sure that you lift it up, and you look, and you're like, oh, and they kind of freak out a little, and you count, and you see what you have. And then you put it back down. So that's the way we have been doing this for a long time. One species, a few species at a time, a lot of effort, see what's going on. So one of the things that we started to invest in is something called environmental DNA. DNA to the rescue. I am not a molecular ecologist. I came to this because of this problem of how do I keep track of species. So the basic idea here is that everything that's alive, including you, shed your cells, and therefore you shed your DNA all the time. So every time, if I touch this right here, I've left my DNA. And if a person comes in with enough of the right things, like a little swab, they can swab right here. And if they have enough genetic information about me, they know that I was here, right? So that's true for everything else that's alive in the world. They're always shedding their DNA, and they're shedding it into their surrounding environment. So what environmental DNA says is if I can go in and grab some of the environment and pull their DNA out, and then I can sequence that DNA and connect it to a species, I know who was there. I know what species were there, I should say. I don't know exactly who was there. For humans, they are scarily able to tell exactly who was there. In this case, all I want to know is what species were there. So the way this was originally developed is from aquatic systems, because you can imagine that if a fish is swimming through water and they're kind of sort of, you know, a little slimy anyway, that they just sort of shed a lot of DNA into the water. So all you got to do is go and pick up a little bit of water, and it has the DNA of a lot of different species from that water. So that was how it was originally pulled through. So this is just a sort of example of that. I think I kind of got that point across. But you can then, I guess the point here is that instead of having to go in and catch every fish or every amphibian or every little aquatic insect, I can go in and just go gloop and grab some water, and I'm done, right? So here's a little bit more diving deep for those of you who are interested. You take a sample out of wherever, in this case water, you filter it through these filters that have tiny, tiny holes in them, and those filters capture complete cells, the organelles from cells, or even the free-floating DNA in some cases. And then as soon as you have that filter, you don't need the water anymore, right? That goes back in. You take that DNA into the lab, you extract the DNA off of the filter, and then you sequence that DNA. You have to choose before you do this what little segment of DNA you want to go for. So you kind of have to choose, like, I want to look at all the vertebrates, or I want to look at the beetles, right? But just from that one piece, then you can sequence all the DNA within that particular region of a genome. Then you get a whole bunch of sequences out, and then you take those sequences and you match them up to what species they came from these global databases on genetic diversity. So we have been building these for about 10 to 20 years. GenBank, the Barcode of Life database. So these have been efforts to just sequence everything in the world. And so they're huge now, and they're public facing, so anybody in the world can access them. So for my little pond in New Jersey, I can take all the DNA that I sequenced out and I can compare it to those big global databases, and they can say, oh, that gene is from Pine Barrens tree frog. Oh, that gene is from a little fish in that area. And I can go through and do all those things, right? You can do these more sensitively on a single species. You have to be a little bit better with the bioinformatics, but the sequencing is a lot easier. So where do we collect this from? You can collect this from soil. You can just go out and grab some soil, extract all the DNA from there, and figure out what is in the soil, like all the little microbes and stuff that are making the soil good or bad, or is there anything missing? But you can also see anything that kind of walked across that soil or relieved themselves on that soil. So the soil is a good repository of a lot of DNA. Another thing you can do beyond just sort of, you know, the water, the things that live in the water, is that there's certain water bodies that attract a lot of terrestrial species, like water bodies in Africa and other more arid or very seasonal places. So you can go and just grab some of that water, and not only do you know what's in the water, but you know everything around it in the terrestrial ecosystem that came to that water body. Now I don't have to fly in an airplane anymore, right? I just have to find those little water bodies and get there. You can look at things beyond fish, things that just use the water every once in a while, beyond just the species that are sort of sitting and wallowing in the water, like water birds, loons, different things like that. So no problem. You just get a little water and you look for birds. That's not too hard. You can, on the other one, is snow. So you can grab a little bit of snow, especially if it's a footprint of something you don't exactly know what it is. You grab a little bit of snow, you let it melt, you filter it through, you find out whose snow track that was. And then finally, these days, we have found that there's enough DNA floating through the air that all you have to do is have a little computer fan in one of those filters on the front and just sit it out in a forest or anywhere else and let it go. And if you let it go long enough, it accumulates, and there's not a lot of DNA floating through the air, so you have to let these things sit out for quite a long time. But all that DNA gets trapped on the filter. And so you can find out, now we have an airflow and aerodynamics piece of this, right? Of what all plants and animals are existing in this, that are going to get caught by this one little air filter. That also completely transforms how we track species in the ecosystem. What this does in terms of solutions is it gets us out of that sort of labor-intensive, one species at a time. You might kill them on accident, or you might have to kill them on purpose to figure out how they're doing, how many species are there, who's there, how long they've been there, what time of the year do they show up, what time of the year to go away, all the kind of questions that you need to know. That you don't have to do that, right? You just grab a little of the environment and look for their DNA. You don't even ever have to see them, right? So I can survey for poisonous snakes, for example, without ever having to get anywhere near them. In my lab, our sort of claim to fame in this world is realizing that there's a whole lot of species in forests or any kind of tree that will leave their DNA on the side of a tree, right? So we were trying to figure out how to take this eDNA technology out of the water and apply it to forests and other areas. So what we decided pretty much over like a beer and Mexican food just south of here is, after a meeting we were at with the state agency, was we wanted to take those little forensic swabs that you can do in sort of the CSI world and giganticize it, right? And then collect a bunch of DNA all at once. And someone, between sips of beer, says, how about a paint roller? And so we tried it. So it works really, really well. So this is us working in Hawaii for a pest insect called the coconut rhinoceros beetle that is moving through the Pacific Islands and decimating all their palm trees. And so they're really interested in knowing when these beetles show up on a new island. And so we look like we're crazy, but this is one of my research staff in Hawaii, and you can see she just has a long painter pole from Home Depot, and then a roller, again, from Home Depot. This is not, and then we go through and make sure we sterilize it to make sure there's no other DNA on it before we take it out. And then you moisten it a little bit, and then you just sit there and paint the side of the tree. Like I said, you look like you're a little bit crazy. But what you're doing is taking any DNA, in this case that a coconut rhinoceros beetle may have left on the side of that tree, and transferring it onto the nap of the paint roller. Once you get the paint roller down, we just take it and put it in a plastic bag with a little bit of DI water and clean it, like what you do at the end of the day when you clean your paint roller when you paint your house. We just do it in a sterile way and scoosh it up, and you get everything off of there into the water. As soon as it's in the water, we can do the filtering part, right? So that's the crazy things that we dream up, right? The other crazy thing that we did was just apply this to those cover boards that I was talking about. So once you have a little microhabitat and things are slithering in and out, the chances that you see them are actually pretty small. But if you just take the paint roller, this is Cat Kyle again, and you just paint the underside of the board, you pick up all the DNA they left under there. So we can sight unseen see a whole bunch of really cool reptiles and amphibians and mammals that kind of hang out under those boards. The other thing we did was we moved this technology into agriculture. So one of the things we realized was that Water mobilizes DNA off the surfaces of plants very easily. DNA is very water soluble and it moves very quickly. And it was a problem for us in terms of trees because every time it rained, a really big rain, it kind of washed all the DNA away, so we didn't have as much luck finding them. So then we're like, okay, maybe we can use this to our advantage. So one of the things we thought of was in an agricultural setting, and this is using tomatoes, but we've been using it a lot for spotted lanternflies in vineyards in New Jersey and beyond in New York, is that you become the rain. So it looks like he's spraying for pesticides, but he just has DI water in the back. Again, you can pick these things up from Home Depot. And then you kind of just wash the plants, and then you have a little bucket that's also sterile, and you catch all the drip water that you just did. And all that drip water has all the DNA that was on the surface of those plants. So we can tell any insect that happened to be on those plants, including the ones that we don't want, like spotted lanternfly, but also the things that we might want, like natural predators and other things that provide ecosystem services for us, or pollinators. The other thing is that in some, especially vegetable farms, small vegetable farms, is that they'll collect all the stuff and then they need to wash it before it goes out and gets sold to you in a little farm stand. They have to follow pretty tight USDA rules to make sure there's no spread of nasty things that you don't want, which works great for us because they rinse everything down with bleach before they do the next wash. But as soon as they put all those tomatoes or whatever, peppers into that wash station bucket, all that DNA is in there. So we just go in after they wash and grab a little water, and then I can tell you every insect that was on that, in that field, right? So those are the ways we're kind of trying to operationalize what's going on. I'll tell you one quick story. So this is, how many people have ever heard of the X Prize? One, maybe a few people. So the X Prize is sort of a consortium of folks who have a fair chunk of money to throw at problems, and they want to collectively push science forward in interesting, commercialized, but other frontiers. So there's a lot of these, and one of them is X Prize Rainforest. And so this is recognition of this problem of every cog and wheel. How do you keep track of biodiversity? They are thinking of it in the context of tropical forests because that's where most of the Earth's biodiversity is. So how do you get into these forests, and how do you know what species are there, and how do you know when they're gone, and how do you know that this particular chunk of habitat is really valuable, so you need to preserve it? It's really a difficult problem. So they wanted to push the technology forward by having an X Prize rainforest. So I got pulled into this, pulled is the right word, by a colleague of mine up at Yale, who had passed through the beginning parts of this competition. They just finished it 2 years ago. And he was doing a whole bunch of really cool stuff with acoustic monitoring and visual monitoring. And one of the key things they set up to make the X Prize hard, and they always set up barriers to make you invent new things, is they said, you have to tell me everything that's in that rainforest, but you can't step foot in it. So everything had to be through robotics. And so he was doing it through getting visuals and getting sounds, but he didn't have an eDNA component. So he came and asked me whether or not we would do the eDNA part. And I, for better or worse, said yes. Because we were pretty far on, we had to, along the pathway, we had to do this very quickly. So we just mobilized the whole lab. The finals were held in Brazil in the tropical rainforest there outside of Manaus. They just boated us out to the middle of nowhere. There was, and then the other part was they're like, you can't use a lab. You're going to figure this out with no lab. No electricity or intermittent electricity. You have to carry everything in and everything out. So how do you get a molecular lab in a boat up the river and then out to this station in the middle of nowhere? That was quite a feat. I'll tell you that sequencing technology has actually progressed to the point where you can actually do that. Little bitty tiny sequencer you put up with your computer. So we got it all out there. We did all the things. We came up with a couple really cool ways of collecting eDNA from drones, and we came in second place, right? Yeah, no. That experience was exciting, frustrating, stressful, and it really did push us in ways that we're still, we're trying to, we've picked up some of those things and are pushing it forward for all the other stuff that we're trying to do. Taking it home to New Jersey, what are we doing in old dirty Jersey, right? So what are we doing here? So we are involved in a lot of projects related to, for example, Pine Barrens tree frog, which is a beautiful little frog that runs around in the Pine Barrens. It's state threatened and endangered. It exists in these vernal pools, meaning they're seasonal pools. They have to be free of fish. They're really tolerant of kind of the acidic waters associated with the Pine Barrens. But the worry, of course, is that as climate is changing and then habitat is changing, we need to hang on to their habitat. So we're trying to help the state figure out where they are now, and then also in the future of like where they might disappear. Another one are corn snakes. This is a little corn snake that sounds so boring. Corn snake, that's boring. They're gorgeous, right? And one of the things that's a problem in New Jersey and around the corn snakes range is that they're so pretty that they get poached regularly to get sold as pets. So if you want to know where to find a corn snake in New Jersey, no one will tell you because they're worried that you're going to go poach them, because any one of these snakes can go for several hundred to $1,000 online. So we're trying to use those cover boards and everything to help the state figure out where the corn snakes still are and keep it kind of quiet, right? So they can manage that habitat and help manage poaching. Another thing that we're doing is there's a whole lot of dams being removed. Maybe you guys are involved in this or are downstream from one, but there are a lot of legacy old dams that don't have a reason to exist in New Jersey anymore. And the Nature Conservancy and the state and other things are gradually going through and pulling those dams out, making those waters free-flowing again. And so one of the things that does is it allows migratory fish that spend most of their life out in the ocean and come up our rivers to breed, to get into more of our rivers to find the breeding area. So this is American shad, this is river herring. These are all an alewife. Like I was in Boston, there's a whole town called alewife, right? It's such an important cultural legacy of the Northeast to have these massive migrations of fish, but they're pretty much decimated because we've just systematically dammed every one of our rivers. So if we can open up the Delaware River watershed, so when they come in the Delaware, they can go up into their breeding areas and still breed. What we're trying to do is use eDNA to show success, because it's a really hard problem politically and otherwise to take those dams down. So if we can help them show that the fish are back, then we can generate some momentum to keep going towards solutions. And the final thing that we're not quite doing yet, but I really, really want to do, are sharks. So I would love to, I don't know if you guys have ever heard of Gotham Whale. So this is a nonprofit group that keeps track of the return of all the whales to the Hudson River and the whole estuary. Have you guys seen the humpbacks and stuff that will come right up to New York City? If you haven't, go look online. So we have a lot of whales coming back, including some really rare whales. So they're having trouble with all the shipping traffic that's happening, coming in and out of the New York harbor. But over the whole, the whales are coming back. So there's this whole group called Gotham Whales. The same thing is happening with some of the sharks. Some of the sharks are coming back. So I want Gotham sharks. Including white sharks, or great white sharks, like the Jaws sharks. They are establishing breeding nurseries like in the Long Island Sound. They get seen regularly now off the coast of New Jersey. So they're coming back. And we have a couple of sharks that are kind of following the climate up here, including tiger sharks. So sometimes like you don't really want to come across one of these sharks. So part of what we're doing is trying to figure out where are the important parts of are there nursery habitats within New Jersey that we need to protect? Where do we see them regularly so that if you don't want to come in contact with a shark, I can tell you when they show up, right? So, and then just in terms of just the, ecosystem stability and what it means to have sharks back here is pretty cool. So, there's like the piece of what I do that's like, don't you want sharks? And people are like, not really. Don't you want to see all the cool snakes? Nope. But for the things that they provide and what we're doing, like there's a piece to both keep the people who don't want to interact with them not interacting with them without having to kill them all, and then keep the stuff that we really want them to do, them still doing, to make sure they're still pretty safe. So I'll end with just pointing you to a couple places One of them is a whole series of work on where's the hope as we move forward. And if you're interested, or if you're interested here, or if you're a YouTube user, or your kids or your grandkids are, then you can point them to Isaiah Hernandez, who on YouTube goes by Queer Brown Vegan. He does a whole series of really nice videos associated with some of the stuff I've covered here, including one interview with Ellen Kelsey, who again is another person, if you're more into reading books rather than watching YouTube, then looking at her book on hope, sort of the evidence for hope. So they don't talk about eDNA in there, but I have drawn a lot of inspiration from the things that they're doing and what they're talking about. And then ending, I think, kind of pointing out the fact that we need these little revolutions in technology, which I've talked about, and there's others, to be able to track how we are being successful in our protection, restoration, and conservation of the ecosystems that we depend on and all the other species that we share the planet. So thank you.
Thank you, Professor Lockwood. You guys already know what to do. Go ahead, sir.
Go ahead.
Thank you. That was a really cool talk. I've done some work in proteomics, okay, which is the downstream from DNA. And one of the things that I noticed when you look in the databases like UniProt and things like that is there's a lot of conserved species that are conserved proteins that are identical across a number of species or only have small changes.
Right.
So, I mean, do you find yourself sometimes when you're doing eDNA, do you find yourself looking, well, it's in this genus, it's not really in this species, and how does that affect what you do?
So pretty regularly. So you just defined one of the problems that we, especially when we're trying to maybe track one particular species, the river herring are a very good example. So there are two species that are here, blueback and alewife herring, right? They both migrate through. They are so closely related that their genetic makeup is so close that with the tools we have, the molecular tools we have, we can't tell them apart. So that's an example of just one example. Now, when you're doing like all the species that you see, like all the fish, for example, then you either are limited by the fact that you don't have enough sequences from all these different species, so it's difficult for the bioinformatics, the information piece to connect, or that you're really only looking for one base pair difference between two different species. And in that case, it's just like you just have to go back to genus. And with the insects, because we really don't have, like the fish are pretty well covered in these big databases, but the insects are not, although the people up at Canada are working their hardest to make it true that's not true, then we often have to go back to genus or family. So it's exactly what you said. We kind of have that problem. One of the things that we're doing on that is we're trying to get enough sequences for all the fish in New Jersey so that we have a New Jersey-centric database with a whole bunch of sequences so that we kind of overcome that problem as much as we can. But yes, it's a definite limitation.
Thank you.
Sure. Zipper? Yeah.
Hi, thank you so much. Very fascinating lecture. I was wondering, with the work that you're doing, have you been able to identify new, or has anyone who's doing this work been able to identify new species, especially mammalia, right? Mammals, because we're so fascinated by it. And in that case, are you able to replicate that species of how it would look or how it would behave and what it's made up of based on today's technology?
So people are just now starting to work on that, because if you have those that little branching tree, the phylogenetic tree, and you know that there's a gene, what the gene sequences are that help define that, then when you get a new sequence from some environmental DNA thing, you can slot it in. And you can say, and some rules, you can say, oh, it's just different enough from everything we know that it might be a new species. So then you can kind of go back, because you can trace and say it came from this water body or this tree. And you can get all the taxonomists out there and say, go look for it. There's something new out there. And so they can do their job a little bit more. So they're not kind of looking everywhere for something new, or we're waiting for an accidental discovery.
So
So
I'm
yeah.
thinking Bigfoot when you told me.
Yeah.
It's like, we're all waiting.
eDNA for Bigfoot. That would get me, that would get me in the news.
Old record.
Yeah, exactly. Hi.
Hi, yeah, thank you so much for this talk. The Adirondacks is also one of my favorite places in the world, and they're officially out of deep freeze season, so time to
Yes.
head up soon. But I was hoping you could help me tease out an observation and answer my questions with this. So I spent about a year in Sri Lanka at a water research institute, and the vast majority of the environmental research we were doing was based in the tropics and tropical climates. And I noted a lot of the research you've done as well is in places like Hawaii or Florida. And even just understanding all it takes to grow a plant is some sunlight and water. So it makes sense, right, that like a rainforest or a tropical humid climate would do well with sustaining life and biodiversity. My first question is that observation correct? Is a lot of environmental, specifically biodiversity research conducted in the tropics? And if so, why on a base level does a warmer temperature sustain life better? Just because in my experience, we weren't getting a lot from like the Nordics or colder climates.
Right. So, yeah, there's a sort of universal rule that most biodiversity is around the tropics. And as you move north or south latitude, that drops off. And we've spent the better part of 50 years trying to explain exactly why that's true. And some of the things that you have mentioned has come up, but we don't really totally know why that's the case. So it's probably a combination of a lot of different things. But yeah, sort of sunlight, because the middle of the tropics get sunlight pretty much steady year round. There's a lot of energy coming in for photosynthesis and everything else. So that's one idea.
Yeah.
And then it creates a lot of different niches or niches depending on your pronunciation across there. So all those, there's like 14 different hypotheses for why most of the Earth's biodiversity is around the tropics. Now, in terms of where all the money goes, it goes, like if you look at biodiversity research and you mapped it out in terms of what we know, North America, Australia, and Europe dominate. Why? Because there's a lot more money shoved into research and environmental things in those areas. Some of those people do work in the tropics. There's a lot of really up and coming, very good tropical biology that happens in those institutions in that area, but it's just not funded quite the same way. So there's a big funding disparity and knowledge disparity because of just funding things. And then there was one more piece of, or did that?
That pretty much answers
Okay.
it, but I was wondering why the warmer, well, I don't know if this is the case, but why on a, I guess, molecular level, like why a warmer temperature sustains life? Is it having to do with energy, or is that a...
Well, so that
I'm not
goes
sure
back
if
to
that
those
makes
14
sense.
hypotheses.
Right.
But I think a strong beginning piece is the fact that as you look at the Earth relative to the sun, right, and the way we circulate through is that the tropics always gets the most sun, right? In between the Tropic of Capricorn and Tropic of Cancer, you have the most sun coming in. So because all life works, well, almost all life works on energy from the sun, then the more energy coming in, you can imagine there's more possibilities for life. And then once you start that ball rolling, so there's more life, then there's more life that can live on the life. And it kind of snowballs from there.
I see. Thank you so much.
Sure.
Hello.
Hi.
I really liked the lecture. It was really nice, and you explained everything in a really understandable way. I've got two questions, if you don't mind.
Sure.
All right. The first one, I'm a really big fan of moths, and you mentioned some stuff about them, and I know they're really hard to classify, and so many of them are undocumented and unnamed and all that. I was just wondering if you like saw anything with that.
So we haven't used eDNA much for moths, I should say, but there's no reason we couldn't. We just need to make sure that we have, like even just looking at that sort of light trap, like can we just use the DNA they leave behind to figure out what was there? So that's possible. We do pick up Lepidoptera, so moths and butterflies, on our paint rollers when we're doing things, and when we look at understory. But yeah, the sort of the genetic resources that are associated that allow us to get from DNA to species is a little sketch. So we could do a lot more related to that. Do you know about Moth Week?
No.
Okay, look that up. So there's a worldwide week devoted to moths. And so if you connect into that, then I think you're going to find a whole lot of fellow travelers around the world who are really fascinated by moths. And if you want to do something, there's a researcher up at Rutgers that works only on moths, and so like doing like field stuff. And so if you email me or something, I'll try to get you hooked up.
That's probably not happening for a while. But thank you so much.
Uh-huh.
And then my second question was, what common issues do you come across when it comes to the eDNA?
It's skepticism because I tell you something's there and I can't show it to you. They're like, really? Is it really there? And some of the questions on like, the problems maybe of connecting a sequence to a species. And so some of that is there. And then especially if there's an action that needs to happen after I tell you that something's there, like when we do the coconut rhinoceros beetle in Hawaii and I tell them, hey, you have coconut rhinoceros beetles, the response, because of the cultural and economic damage that they do, is to actively manage, including applying pesticides and other things, or taking down the tree. That's a big action, right? And so doing that action based on a DNA signal makes people very uncomfortable. So, we kind of work around that by thinking about eDNA within a decision tree, because you need more evidence maybe to make those decisions. So we kind of get into that piece. And the other is like, the other piece is skepticism. If it's so easy to move DNA from one place to the other, how do I know that you didn't move it there? So we have a lot of controls in the process that should come out negative. And if they don't come out negative, we know we have a contamination. So there's things to do, but I have to like preach it over and over and over again and tell people, especially skeptics, it's like, it's okay. We have steps along the way to make sure we're not doing something stupid. And we're not trying to say that this eDNA signal is the end all be all, there's other things you need to take into account. But that's a great question. Thank you.
Thank you.
2 quick questions from Zoom, if I may. First one is, have you ever considered, instead of the cloth roller, a sticky lint roller? And second question is, with so much DNA and so many different species in that DNA that you're cultivating, how do you pinpoint a particular sequence and amplify that? How do you pick the one that you're looking for?
Right. So what was the thing? What was the first one?
Sticky lint roller versus the cloth roller.
Oh, like a lint roller. No, we haven't talked about a lint roller, but I can, if we do it, then I'm going to say it all happened here. We've done other things, which I'll talk to you about in a minute. But anyway, so there's other strange things that we thought about. And then how do we know what species it is? So what you do, sort of to get into the weeds a little bit, is that of course you have an entire genome associated with each individual, and the species has their own genome. And what you're trying to do is pinpoint a small section of that that's very diagnostic for that group of species. So there's a fish part that looks pretty good to distinguish all fish. So, and these are folks that do this, like I don't do this, but they do this. They use bioinformatics, they use existing sequences, and they go, oh, that looks like a really good sequence to tell the difference between all the fish. So then they develop sort of the molecular tools, the probes and everything that cut the DNA along that area, and then you just sequence that little bit. So it's very
A flow chart, if you will, of
it's a flow chart, but you're purposely looking for a chunk of DNA. And eDNA, the chunks that are left are pretty small. So we're talking about 200, 300, 400 base pairs out of millions sometimes. So we're looking for that little slice that can tell us, like this slice can tell you the difference between all the fish.
Quick things. 1, you're an awesome science educator.
Oh, thank you. You know why? It's because when you teach for a long time in front of a lot of people who don't think they care, it's trial by fire, so you get a little better at it. Yeah, go ahead, sorry.
Okay, there's a quote from Lauren Isley. If there's one substance on Earth in which magic is to be found, it is water. So that plays on 2 levels. The material itself, which freezes and floats, only 2 of those that do that, right? Some other things, but everybody has talked about climate change for a long time, right? Why aren't more people talking about the 6th mass extinction? Like, that's quantifiable. That's something that's like, go ahead and try to argue with it, because you can't. Like, the amount of biodiversity is ridiculous that we had 200 years ago, and it's,
Disappearing.
yeah,
Yeah.
the passenger pigeon flocks that were 300 miles long and a mile wide, like, gone, like stuff like that.
So I think there's 2 reasons we don't think about it. One is that the way we have thought about biodiversity is emerged from the 70s environmental movement, right? And so it was always given as more of an aesthetic, like, what are we doing? You're doing bad things. What are you doing? You know, and not necessarily thinking about it as, we can't survive without this. You know, and so the question is, how much loss can you have before you really feel it? And so there's a shift in focus, like the International Panel on Biodiversity and Ecosystem Services just came out with a nice thing on the connection between business and biodiversity. So we're trying very hard to make those connections so it's more clear, so it's not just like a nice thing if you can have it, but instead it's something central to everything that we do, kind of like how climate is, right? And so we're working on, I think it's been sold as that in certain ways, and then as a nicety that you want to hang on to. But it's too easy when it's a nicety to say, but I need to make some money, or I need to find something to eat, or I need to do this, and instead try to integrate it and be like, look, you can do that, but there's a point at which it's going to become harder, and we need to be aware of what that is. So I think that's part of it. We're kind of challenging that narrative and what's going on without losing that heritage of, you know, sort of the 70s sort of environmental movement. So that's part of it, yeah. Yeah,
It's-
I know. And also the other part is that it's like this shifting baselines idea of, like, when my grandfather went fishing in West Virginia or something, his experience of nature around him was very different than when I might go through those same areas on a hike. But I can't see that. I don't feel that, right? We didn't record then. He didn't tell me what it was like, right? And so I can't, and so to me, I can only judge the changes I've seen over my 50-something years. And so there's another part that we just lose that, and we don't really understand how much we lost.
Yeah. Okay, thank you.
Thank you.
Hi, thank you
Hi.
so much for this talk. I'm really into plants, and I was wondering if you can use eDNA to find plant life.
Yes, you can. So it's well developed for aquatic plants because it's all started in aquatic world. But especially with air DNA, because it catches pollen. And so we're kind of getting a much better sense of what you can do in terms of plants. And most of that's through this emerging idea of air DNA. And then one of the really cool things is that when they first showed that air DNA really works is so there's all these EPA monitoring stations, places for pollution. And then they have filters on them, and then they pull out from those filters like all the pollutants. Well, someone grabbed some of those and ran all the DNA. And so all those filters that they were using for EPA air filter and making sure our air is okay also has a biodiversity signal in it. So the question is, like, how long does that last? Can we go? They keep those going back decades. Like, can we go back decades and figure out and get at that question of how things have changed? But yes, but mostly through pollen getting moved around.
Mhm. Yeah, also, you mentioned the plant that there's only 300 of them,
Yeah.
like the
Cycads.
cycads. If you could use those plant DNAs and find the pollen, would you be able to replant them?
Yeah, absolutely. So you'd have to go and get somebody who'd do all the bioinformatics, which is not me, and figure out what part of the genome you could say, okay, from this part of the genome, I can tell all the cycads apart. And then you'd have to know, okay, when do they release pollen and do other things so there's enough DNA for me to capture it somewhere? And if you could figure those things out, then you can deploy them anywhere you want and figure out where all the cycads are. And then once you know where they are, you also know where there aren't. And then with a little more information, you can say, oh, that's a place where I should go in and restore.
Oh, okay. Thank you so much.
Sure. Hi.
My question has to do with the every cog in the wheel part of your talk.
Okay.
I once read a book titled The Mosquito, and it was written by a historian, and he traces through events in history in which the disease caused or transmitted by mosquitoes affected historical events.
Mm.
He also claimed that of all the species in the world, the mosquito is the only one that plays no useful role in our living, our lives. How would you respond to his claim?
So my youngest son says the same thing. So when you grow up the kid of a biologist, you have to like automatically know a lot of biology. So he's a computer science kind of nerd, but he will talk regularly. He's like, we don't need mosquitoes. What do mosquitoes do? It's like they don't have any role in the ecosystem. So maybe that's a cog we can do without. I don't know. You know, that's a good question. I mean, they do serve as a food source. They are part of the food web. So when if we lost them all, would it have a huge impact? Good question.
There
Good
are
question.
efforts to
rid of
reduce
them
their
all.
population.
Yes, so and some of those are spraying a lot of stuff that we don't want because it gets rid of a lot of other things. Some of them are more technical and interesting.
Yeah, they're
Like-
biological means.
Biological means you genetically modify a few of them and then they mate and then nothing happens,
Right.
the sterile males. There's some really cool ways. And there are some places in the world, including Hawaii, where I do not want mosquitoes because they vector diseases that the native birds there are very naive to, and they die off in droves. And so, and they were never on Hawaii. So I'm all for knocking back some of the cogs. Getting rid of them? I mean, I guess I would say I wouldn't be ethically totally in favor of that, but I get the point.
Okay, thank you.
Mm-hmm.
Thank you so much.
Sure.
My question may be related to a little sci-fi,
Yeah.
but since you're in this field, maybe it becomes reality. All the DNA samples that are being collected for those species, especially which are being threatened or being extinct, using the technology advancements, do you see that would it be possible to recreate those species back to life?
Probably not. So because we really only have one segment of their genome, right? And so unless you had the entire genome, there's really not much chance. And then even then you have to create an organism that has all those genes, and you can't do that from scratch. And so when you see things about de-extinction in the news, I mean, what's happening is they're inserting a good chunk of DNA into an existing species. So is it really the thing that went extinct? No, it's some sort of hybrid between that thing and the thing we still have. So you really can't go back, even with all the DNA we have. Although, yeah, I mean, it would be nice if you kind of could, but unfortunately, it's not possible. Yeah. Thank you.
Thank you so much.
And our last two questions.
Great talk. Thanks for being here.
Sure.
Curious how robust this eDNA process is for looking at the numbers of the individuals of species that were in the area and the recency of them being there. And if it's not so robust, do you ever expect eDNA to take over as the primary method of tracking species compared to the usual ones?
Right. So the recency varies depending on what kind of environment you're pulling it from. So we know from a lot of studies in aquatic systems and our DNA on the side of trees and stuff is that it's usually around for about 2 weeks. That's it. It just degrades because of UV and microbial activity, and just every time, once you leave DNA out, it just starts to chop, right? And so once it chops so small that our little chunk is no longer able to, you just can't get that big of a piece, then that signal it just goes away. Now, under a cover board where it's not getting a lot of UV, I'm not entirely sure. It could last for longer, months maybe, but but unless the DNA gets attached, so another thing is DNA is polar, it has a polarity. So it can get stuck to soil. And so if that soil gets buried, then it can last for tens of thousands of years. And sediment of lakes and old things. So you can do soil cores all the way down, and depending on how far down you go, you go back in time. If you take that DNA out, you can get an overall kind of idea of what used to be there. So we stay away from soil for that reason for some of the applications that we use, because we need to know it's recent. So but on the other hand, if you don't want to know it's recent and you're looking then you should go to the soil and the sediments, because you'll get a deeper. So it's really complicated in very interesting ways. Can it tell the abundance? So that's been, right now, no problem, it's there or not. But we're getting much, much better at figuring out, little pieces of what the DNA looks like. Is there methylization of the DNA? Is there just ratios of different parts of the DNA that tell us how many individuals are there? And we can also now say, oh, this DNA came from a male, this DNA came from a female. So we're getting much better at getting past just there or not there. Will it ever surpass everything else? Probably not, because even with that, there's probably pieces that we can't quite figure out, that we really need the little cool geolocators and things that we can attach to them and follow them around that we can't get. But it's a revolution. It's truly, it's taken over marine fisheries so that you don't have to kill everything to figure out how many there are anymore. You just grab some water. And I see the same thing happening in forest health and maybe in agriculture. So it's moving, but it's sort of like AI. It's like, is AI going to take over everything? No. But will it transform how we do things? Yes. Yeah. Thank you.
Fantastic. Thanks.
Thank you.
Hi.
That was a very interesting talk. I just had one question. So when you're taking samples from liquid like air or water, I understand that it's more or less homogenized, so you can get a pretty accurate representation of the biodiversity within like the area. But for something like terrestrial samples, I was wondering how granularly do you have to like take the data from a certain area to get like an accurate representation of what lives there?
Oh, that's a great question. So, like I'll take on the side of a tree, right? So there's some DNA that will probably get into the air from there, but it's not a lot. So what we have found we have to do is cover a lot of surface area. And we also stay away from the soil for the reason that the DNA connects to it. We don't know if it's a recent signal. And also because it's super localized, and processing a whole lot of soil is totally a pain. But when you see Kathleen out there rolling that tree, we're like, how many trees can she roll before we lose the DNA signal? We did like, how many rollers can you roll? That was a whole paper we did. How much surface area can we cover? And what we found is that there's a point at which you start, all the DNA that you collected from the prior tree starts to get redeposited on the tree that you're doing now. So there's a limit to what you can do. But the question absolutely is, how much surface area can you cover? And the other thing is taken into account, what if you're looking for different species or maybe a particular species, it's like, what is their natural history? Where are they likely to hang out? Because where they're hanging out is where they're going to leave a lot of DNA. And for insects and other things, it's also, where does their poop go? Because we're basically kind of like trying to track insect poop in a way. And so when that helps you narrow down the giant forest into a few trees, so that you use those tools, because it absolutely does not mix. Like in water, it mixes pretty well, not completely. But in the terrestrial world, it gets deposited and it stays. And so the game turns around. That's a great question. Thank you.
Thank you.
Please help me thank Professor Lockwood one more time.
Thank you all.
And I'll see you next week.